So thank you, Sam. You actually just gave I think the line I was about to give, which is that, so now we can make motor neurons. You've heard about this. Now we want to talk about what can we actually do with the motor neurons. And so, for example, we want to go from the stem cells to forming functional neural circuits or modeling some disease processes. But I want to just take one moment, or probably several moments, to just point out one thing, that we talk about motor neurons, but motor neurons are incredibly diverse. And this is because we have diverse motor tasks. And I use the example here of Michael Phelps, the swimmer, because I think this image kind of captures that motor activities are quite broad. There's not only just locomotion, but we're also talking about control of breathing. There's control of visceral functions. And one thing that's very convenient for studying these processes and the formation of these circuits in a developmental context is that these form very early in development, such that at very early stages of life, at this point, 10-month-old son had he was endowed with the same motor control functions that Michael Phelps has. It'll take him some time to get there, but the core circuits are there. So in line with this diversity of motor activities, there are many different types of motor neurons, and these are arranged in particular positions along the developing body axis in line with the different types of muscle or visceral targets that are involved. And these, of course, are subdivided by proteins that organize the body plan, the Hox genes, much as the rest of the body structure is also subjected. And if one cuts a cross-section through the spinal cord, one can appreciate that there are different types of motor neurons that they occupy specific positions within the spinal cord, and this is also matched to their peripheral projections. So motor neurons that are in a lateral position tend to project to more distal targets, and in this case here, looking at limb levels, they innervate the limbs. Whereas those that are more medial are projecting to more proximal targets, such as the axial musculatures of the vertebrae, as well as respiratory muscles along the body trunk. And so one of the real questions that we have as a field is trying to understand how we can take stem cells and how we can walk them through these developmental processes to create the diversity of cell types that we would like to be able to study, because just one particular type of motor neuron may not be able to do it all. And so in addition to this breaking down of motor neurons into what are called motor columns, because they extend along large segments of the body axis, these motor columns can be further subdivided into what are termed motor pools, which are defined as the motor neurons that innervate a given muscle within a target region. So, for example, there'll be multiple muscle pools that will innervate different muscles in the dorsal limbs. And another point I just want to just bring up is that these different motor neurons have diverse activities, and even at the electrophysiological level. They have clearly different innervation capacities, and it's also been noted that in terms of pathologies that there's different disease susceptibilities. Certain neurodegenerative disease like ALS tend to affect the distal projecting motor neurons more than the proximal ones. SMA, it's the reverse. So how can we approach this? And since the session is largely focused on the transcriptional effects, I think that this is where we can really start. And really through the pioneering work of Sam Pfaff and many others, Tom Jessel and his colleagues, we've come to the appreciation that one can define many of the characteristics, the overt characteristics of different motor neuron subclasses based on the combinations of transcription factors that they express. And this is just showing immunohistochemistry and then just a lineup of some of the different markers that are in there that define these different colored populations of motor neurons. And these, as Todd just indicated, that these aren't just markers, these are actually functional proteins. And by simply changing the code, essentially, by taking away one of the transcription factors, and this is some work that we did a few years ago with the FOXP1 transcription factor, which defines the motor neurons that innervate the limbs. If you get rid of that transcription factor, you get reduced limb innervation, and the phenotype of the motor neurons changes something that looks much more like those that innervate the diaphragm. And so this really says that these are useful tools for changing the properties of the cells. So let's talk a little bit now about the stem cells. So first thing, one thing that I think is very important to appreciate is that as Todd and Sam had also introduced the concept, it's a lot easier to study things in mice because we have the genetic tools, we have great protocols for differentiating cells, and we can study the whole animal. We can actually study motor behaviors. But we're interested, of course, in trying to apply this towards human. And so one of the things that's a big challenge for the field right now is kind of catching up with the human biology to really see, how much of what we study in the mice actually relates to the human. I can just say that this is something that mine and other labs are interested in, and if one just looks through the immunohistochemistry at human fetal development and mouse development, there's remarkable conservation. The same markers are there, the same organizational principles apply. Now, when we start looking about stem cells, this is where things get a little bit challenging, in that we have methods that we can make motor neurons with pretty high efficiency. Mostly these are based off of the retinoic acid plus sonic hedgehog paradigm. And what's very good is that you can make lots of motor neurons with those things, but you don't generally make the diversity of motor neuron subtypes that one would see in vivo. So in both human and in mouse embryonic stem cells, when differentiated under these methods, they tend to form the motor neurons that look much more like the medial motor column, the ones that innervate the vertebrae, and not those that innervate the limbs. And of course, we're interested, and in another property is that where you gain that property, you lose the property of limb innervation. And so people have gone on further to show that if you transplant these embryonic stem cell-derived motor neurons into, say, a developing chicken spinal cord, there's preferences in what these motor neurons will innervate. They tend to innervate the axial muscles. They do not innervate the limb muscles very well. So one of the things that we're trying to get at is how can we expand this diversity using some of the transcriptional programming approaches. So I'm just going to walk through some of the experiments that we've done today, and just to show you that we can do this, that it's a viable approach, and that it actually makes a difference to the motor neuron and what it can do. So we, as two of my graduate students, Katrina Adams and David Russo, so David had done studies looking at the role of FOXP1 during development, and he had generated a transgenic mouse in which he expressed FOXP1 in all the motor neurons under the control of the HB9 promoter. And we saw under those conditions that there was an expansion in limb-innervating motor neuron production. And we thought that we should test whether or not these same culture conditions in which we weren't getting limb-promoting motor neurons, limb-projecting motor neurons, we can now see them coming from these stem cell cultures. So the experiment is we take the embryonic stem cells. In this case, the control is just the HB9 GFP's control line. We differentiate them using the methods that Todd introduced. And Hank introduced, and we can get, as the 30% efficiencies, we can get large numbers, though, still, of these HB9 GFP positive motor neurons. And when we panel them with these different transcription factors that demarcate different motor neuron subtypes, you see that, as I just indicated, they express ones that look mostly like the medial motor column. Just by simply putting in the FOXP1 transcription factor, we can change this profile quite dramatically. We start to turn on markers that are characteristic of a subset of the lateral motor column, all the lateral motor column, something called LDH2, and you can see that there is a suppression of the medial motor neuron fate at the same time that we're getting this big increase in the limb projecting phenotype. So based on the transcriptional information, or at least what we can see by just looking at these transcription factors, we can see that there's a clear biasing now of the development towards the limb projecting motor neuron fate. So we wanted to go a couple steps further to show that this really actually had some more meaningful outcome. So, and I also should mention that we were able to see the same sort of things by using viral methods to put FOXP1 into human stem cell-derived motor neurons. So I think that this is something that will work on humans as much as it does on mice. So let's look a little bit closer at the FOXP1 expressing motor neurons. So under control conditions, when we differentiate these things, we went looking for what are termed the Motorpool markers. So these two can be defined by different transcription factors. And when you make motor neurons in the control population, we very rarely see signs of overlap between GFP and any of these markers, which are here in red. But this is dramatically increased now when we've got FOXP1 expressed. So this is telling us that we're able to convey a sense of competency of the stem cell-derived motor neurons to turn on these different motor pools. We still don't know much about how these motor pools are controlled, but this essentially opens the door to say that the cells become receptive to perhaps environmental signals that affect those characteristics. Another characteristic of limb-projecting motor neurons is that they have to be able to navigate to their appropriate targets. And this is something that's been very well studied in the motor neuron field. So we know that there are axon guidance signals which direct the axons in their growth going to different parts of the body. So perhaps the most famous of this, and this is something that Sam Pfaff has contributed enormously to, is our understanding of how motor neurons that innervate the limbs know to go to the dorsal versus the ventral limb. And this is regulated through the expression of ephrins, which is the ligand, and EPH receptors, which are the receptors that that affect axon guidance decisions. And these subpopulations of motor neurons express the EPH receptors that allow them to avoid, say, the ventral limb. And if we look at one of these, FA4, we can see that by converting the motor neurons into limb-projecting phenotype, they show this nice upregulation of EPHA4. And with some studies that we're doing currently in collaboration with Archer Kania's lab, we're interested in trying to use this now as a system where, because we can generate large numbers of these cells, we can do some biochemistry on how motor axons respond to axon guidance signals. And this is just showing you with the HB9 FOXP1 cells, if we just expose them to in culture into a soluble FC molecule, there isn't much change on their axon projections. Whereas if we now dump on FA4, or FNA4, we can see this rapid collapse within 30 minutes. So it's a really nice system to be able to study some of the dynamics of axon guidance. So we can see that there are changes in the way that the cells can respond to axon guidance signals. What about their target selectivity? So we've done assays in which we take the stem cell-derived motor neurons and we put them into culture, and we put them aside either axial muscles or ventral limb muscles or dorsal ventral muscles to ask whether or not they have any preferences for the way that they project. And when we do this, what we find is that the control motor neuron population is consistent with their medial motor column identity, have a preference for the axial muscle, whereas the ones that have now been programmed with FOXP1 go the other direction. They're inclined to innervate the dorsal limbs. Another test that we've used, which is, I'd say, a lot more stringent, is to take these stem cell-derived motor neurons and now to put them into a developing chicken embryo and to ask, where do they go under that environment? Are they able to, A, integrate into the spinal cord? Do they have any ability to project one way versus the other? Do they make any synaptic connections in the end with muscle cells? And so what we do is we transplant in clusters of these ES-derived motor neurons, either control or FOXP1 expressing, and then follow their trajectories based on the GFP expression over many days of development. And what we find is that when we do this experiment with the control cells, very much recapitulating the results of others before us, that there is a predilection of the cells to innervate going north here, which is going towards the axial muscles. And very few start to go down towards the branches that are going to innervate the limbs. Even after 3 days transplant, we can see that there's a skewing in the FOXP1 expressing line where there's a lot more axons that are going down into the limbs. This gets accentuated as you go look further and further in development. And we've quantified this by just making different arbitrary marks along the trajectory of a motor axon from out of the spinal cord going different directions here. And what we find with the controls is that we never find a stable innervation going out into the distal limbs. Whereas in the HB9 FOXP1 expressing cells, we now see that in most of the embryos that we do these transplants, there is over 60% of them show some distal limb innervation. And when they get there, they are actually able to form neuromuscular junctions with the limb muscle tissue. So this suggests that they're making some bona fide connections here. And we also find that they receive presynaptic inputs. If we go back looking in the spinal cord, we can see that there are some presynaptic markers that are organized around the soma. Okay, so that's how we can change the phenotypes of the cells. I want to change gears for the last few minutes just to talk about now what are some of the things we might be able to do with these cells. So one of the things that we're particularly interested in is trying to use this as an assay platform to try to understand the basis of disease. Animal models are great for many things, but in the end of the day, we'd like to be able to design assays to study particularly human disease pathologies, and also to try to use this potentially as a platform for drug screening. So just want to point out one thing that we think about with neuromuscular disease is that it's not just the motor neuron in many cases, but it's something to do with the way that the motor neuron is functioning. And this could be either through changes in the presynaptic inputs to the motor neurons, as I think Sam had alluded to earlier, or some breakdown in the communication of the motor neuron to its muscle target. And when this happens, when you start to have denervation, this really leads to a cascade of things that can affect motor neuron health and survival. And so one of the things that we've been interested in is trying to test out the waters to see whether or not this could be a viable system for trying to study neuromuscular disease, starting with stem cell-derived motor neurons. So a couple years ago, we just did a proof of principle experiment where we just took stem cell-derived motor neurons that were expressing HB9-GFP, and we co-cultured them with muscle satellite cells, which can form these nice multinucleated fibers which have the ability to contract spontaneously. And just to ask whether or not they could form neuromuscular synapses, and they did. They have bungarotoxin, which binds to the acetylcholine receptors. We've also looked at other pre- and post-synaptic markers. They all cluster around these sites of contact. And most importantly, we can do neurophysiological recordings from these preparations, where we can use electrodes to stimulate the motor neurons, use another electrode to record for the muscle cell that's connected, and see that there's paired activities, so that when the motor neuron is stimulated, we see a response in the muscle cell. This is very classic neurophysiology for the neuromuscular system. And importantly, this is all going through cholinergic neurotransmission. We can block this with drugs that block cholinergic activities. But this is a very, this is sort of the gold standard system, but it's very slow and it's hard to get population statistics on these sort of things. So we've been turning to using the new optogenetic methods to try to to increase the throughput of this. And for those who aren't familiar with optogenetics, what this is using light-activated ion channels that you can put into neurons to stimulate their activity just simply by shining certain wavelengths of light. And you can measure muscle activities in the same way with calcium indicators. So just to show you that this system works, we do co-culture of the motor neurons and muscle, and this is just showing you when the light flashes here, this means the light stimulating the motor neurons coming on, and you can see that this works, and we can get stimulated activities. We can also see this in the same sort of preparations, induced contractions of the muscle itself. So it'll take a second, but you'll see a light flash, or the yellow thing will come, and you'll see a big contraction there. And these are all blocked by the same drugs that block cholinergic transmission. So how about this in a disease context? So the disease that we decided to test this out on first is SMA, because it's a genetic disease. We know that the actual disease, gene that is mutated. It's a gene that affects RNA processing, but we don't really understand how it has an impact on neuromuscular activities, and that's something we're interested in figuring out. And I just want to show you just some of these are preliminary findings, but I think they're rather interesting. That using the same sort of assay with controls, and I can speed through this quite quickly after the first few. We have a little bit of time where we're showing activity without any stimulation. Now there's stimulation. And you can basically see the same sort of regularity in activities. When we do initially the co-culture between the motor neurons and the SMA, these are SMA motor neurons. These are from a mouse that's been designed to. So you can see here now the light is coming on. There's no activity for a while, and then occasionally you'll get a flicker that will come up. So these are from a transgenic mouse model of SMA that's been made. And so initially in the co-culture, we actually see that there's hypoexcitability. They're failing to stimulate muscle activities appropriately. But this phenotype can evolve over time. So if we look a couple of days later, the same sort of cultures, what we find is that this profile changes quite a lot. The control doesn't change almost at all. So when the light comes on here, you can see that there's paired flickers of this calcium indicator, very much in line to the stimulation, which are these blue lines. But here now with the SMA ones, what you're going to see is our flickers before we start to even have any activity of the motor neurons. And we actually will see, you can see there's a kind of like a pulsatile contraction. So you see that there's irregularity in this activity. All right. In the last slide, we want to know now, like, is this actually also going to be holding true for human cells as well? And so we've done the same exact experiment using human stem cell-derived motor neurons and using SMA, iPS cell-derived, SMA patient iPS-derived motor neurons. And what we see here is the same sort of things where the control ones, they're not quite as good as the mouse motor neurons. We don't think that they've matured quite enough in culture, but you can still see that there's regularity, stimulus-evoked activities. Whereas in the SMA cells, we actually find a kind of an interesting phenomena, which is that there's all this crazy spontaneous activity of the muscle cultures around the motor axons. And we don't really know yet the significance of this, but it's something that we really are struck by. And this is using human muscle cells, by the way, instead of mouse cells like we've used in the previous experiments. And this is what the activity profile looks like. So we can see that there's clearly some hyperactivity. So to wrap up, what I've told you in the first part is that motor neuron diversity is quite extraordinary. And because we know so much about the developmental biology, we have an appreciation of what are some of the factors that, that contribute to that diversity, and that thus gives us an entry into how we may be able to manipulate those activities to do things, to make particular types of cells. And what I showed you is that most protocols that people have used to date tend to be biased in the types of motor neurons they produce, but we can unpick that bias simply by using these transcriptional profiling approaches, or sorry, transcriptional programming approaches. And the same principles that we're using to make limb-projecting motor neurons presumably could be used to make other populations of motor neurons, particularly those that innervate, say, the diaphragm and those that innervate visceral organs. And the two take-home messages that I want to say about the second studies that we've been doing is that you can use the stem cell-derived motor neurons to model aspects of neuromuscular synaptic disease. And in the case of SMA, we believe that this is going to be a progressive defect where at one point in time, it may look like hypoexcitability, but over time, it may have changed into something more like hyperexcitability. And this gives us some clues as to what the underlying defects may be in the cells, and that's something that we're really interested in exploring over the next few years. So with that, I'd like to just acknowledge the people who did the work. Most of this was the work of a single graduate student, Katrina Adams, and the second stuff with the neuromuscular culture, that's been the work of a postdoc in my lab, Ken Yamauchi. I'm grateful to CIRM and other funding bodies for supporting this work, and thanks to our collaborators. Happy to respond.
One question. So, you have the best question?
I hope so. You've got a monopoly of questions. You three. I mean, I'm just taking the place out. I'll give you one. Thank you, Roger. I was curious, when you do the ventral engraftment of the exogenous stem cell-derived motor neuron precursors, is that a permanent integration into the nervous system? Do they, you know, when the sort of the fine structural sculpting of the motor pool at the level of synapse competition on the muscle fibers, do they participate and do they stay?
Yeah, it's a great question. I wish I could say that we've been able to carry these things out into adulthood. It's actually difficult to keep these transplanted embryos alive long enough to really start looking at those very distal things. What we can say, though, is they are forming neuromuscular synapses based on all the sort of markers. We've done a few experiments where we've put channel rhodopsin in to try to look for evoked activities in the muscle cells. We can see the clustering of presynaptic molecules around the motor neurons within the spinal cord, and they tend to sort, like the FOXP1 expressing ones tend to sort out to the lateral sides of the spinal cord. So they seem like they're migrating to their sort of home area. So we can't say that like it's a complete integration, but all the signs that they have the capacity to at least start that process, they're all in place.
So they are corticospinal targets?
Well, those are most of those come in like after birth, and so that's very difficult to study in the chick system.